Hello everyone, myself is Dr. Sharad Giramkar, working in PDS Anna Sahib Magar College, Hadapsar, Pune, India. In this video, I will explain Introduction to Cell Division, Stages of Cell Cycle, Stages of Mitosis, and Significance of Mitosis. Dear friends, please keep watching till the end of this video, and if you like this video, subscribe my channel, press like button and share this video to your friends. Now, introduction to cell division. Cell division is a process by which cell divides into two or more daughter cells. There are two types of cell divisions, namely vegetative cell division and reproductive cell division. In case of the vegetative cell division, the cell division takes place in the somatic cell. That cell division is called as a mitosis. And reproductive cell division, it takes place in the gametic or the reproductive cells, and this one is called as a meiosis. Today we are going to concentrate on the mitosis. In case of the mitosis, before entering into the mitosis, cell enter into the cell cycle. What is cell cycle? A series of changes of sequential events occurring in the life of dividing cell is called cell cycle. In case of eukaryotic cells, the cell cycle is divided into two major phases. Two major phases, that is interphase and the mitotic and or meiotic cells. For example, this one is the total, this one is the interphase, and this one is the M phase, called as either mitosis or meiosis. In case of the interphase, it is also called as a preparatory phase because in this phase, cell grows and makes a copy of its DNA, RNA, enzyme. We'll discuss one by one. Interphase is divisible into three subphases, that is G1 phase, S phase, G2 phase. G1 phase, also called as a first gap phase, in which the cell grows physically larger and make high amount of protein molecules required in later stage. More cell organelles are produced and the volume of cytoplasm increases during G1 phase. If the cell is not to divided again, then that cell enter from the G1 phase into the G0 phase. G0 phase, it is a resting phase in which cells exit a quiescent stage. If there is a continuous cell division, then cell enter from G1 phase to S phase. S phase is the phase of synthesis in which the cell prepares its DNA. So the chromosome number is double through DNA replication. So therefore it is called as a CS phase, or also called as a synthetic phase. The another phase is G2 phase. So in case of the G2 phase, it is a second gap phase in which cell grows more and prepare proteins and cell organelles. The cell begins to recognize its content in preparation of mitosis. Then after the G2 phase, then actual, we can say it is a M phase takes place. M phase, it may be mitosis or meiosis. I earlier discussed about that M phase, these are of the two types. Whenever there is a presence of the somatic cells, then it's a mitosis. One cell divided into the two daughter cells. Whenever it is a reproductive cell, then it is a meiosis. One cell divided into the four haploid cells. Now in case of the mitosis, it is a process. Mitosis or the M phase, it is a process of cell division in which each cell divides into two daughter cells. Each daughter cell is having the same number and kind of chromosomes as the parent nucleus. The M phase is further divided into two phases. So it is a karyokinesis and cytokinesis. Karyokinesis, it is also called as a division of nucleus, and cytokinesis, it is a division of cytoplasm. In case of the karyokinesis, nuclear division, it is of the four subphases, that is prophase, metaphase, anaphase, telophase. These are the phases of nuclear division called as a nuclear division or the karyokinesis. And cytoplasm, then it ends into the cytoplasmic division called as a division of cytoplasm, in which actual cell divides, nucleus along with the cytoplasm divides into the two daughter cells. First of all, we'll discuss one by one. Prophase. What are the characteristics of prophase? These are: chromatin fibers undergo condensation and appear thick. What are the characteristics of prophase? Prophase shows following characters, in which chromatin fibers undergo condensation and appears thick as compared to interphase. The two pairs of centriole move toward the opposite pole of the cell, and aster formation begins in the animal cell. The chromosome begin to migrate toward the cell center. Nuclear membrane disappear. So nuclear membrane is completely disappear. Aster formation start. Condensation of the chromosome. These are the characteristics of metaphase. Then next phase, subphase, is called as a anaphase. In case of the anaphase, the paired chromosomes in each distinct chromosome begin to move apart from one another. These are moved apart from one another. It's due to the contraction of spindle fibers. Then, as the spindle fiber contract, the daughter chromosomes move to opposite poles. The daughter chromosomes appear either L-shaped, V-shaped, or J-shaped. This is the sign of anaphase. Then telophase. In case of the telophase is a final stage in karyokinesis, in which as chromosome reach the cell poles, the disintegrated nucleolus and nuclear membrane reappear. Thus, dividing cell possesses into nuclei, each nucleus with equal number of chromosomes, like parent cells. The chromosome begin to expand as like interphase. The mitotic spindles get disassembled. Now, cell is ready for the synkinesis. In short, one cell consists of two nuclei. For example, this one is the one complete cell, and that one complete cell consists of two nuclei, in which nuclear membrane appear. Internal set is a condensed chromosome. These are get disintegrated, etc., etc. And after that, cell enter into the actual cytokinesis. In case of the cytokinesis is the division of cytoplasm of dividing cell. In this process, one cell physically divides into the two daughter cells. In case of animal cell, cytokinesis takes place by means of formation of furrow. So it is a groove-like structure or the furrow-like structure formed in the over the that particular cell plasma membrane of dividing cell. The furrow deepens into the cell till the cell divide into two daughter cells. The two daughter cells contain equal number of chromosomes as like parent cells. Now, the next one is significance of mitosis. Mitosis is result of formation of two diploid daughter cells into identical genetic material. Mitosis is responsible for the growth and development of embryo from the zygote. Mitosis is so important for cell repair. The cells of upper layer of a body, inner lining of gut and other organs are being frequently replaced by the process called as a mitosis. In case of plant cells, mitosis helps in vegetative propagation of crops, formation of new plants, new plant parts, and the flippering of damaged part of the cells. This is about the mitosis, cell division, cell cycle, then the phases of mitosis and significance of mitosis. Thank you.